Okay, I'm going to record. All right, so we're going to this week look at complex patterns of heredity. So last week we looked at basically just Mendelian genetics, what it's called, where there's dominant and recessive. Does anybody have any questions on basic dominant and recessive Punnett square stuff that we did last week? Okay, well, the stuff we're going to look at this week is more complex stuff where it's not just dominant and recessive heredity, where there's some other things that happen. And so I'm going to start with, here's the list of things that we're going over. And we're not going over everything today, but here's a list of, so the first one there, dominant allele patterns. That's not the dominant recessive heredity that we've already looked at. We'll talk more about that later, but it's when, if there's a dominant allele at all, then it's always present. And like I said, we'll discuss that along the way here. But today we're going to focus on the second and the third on the list there, incomplete dominance and codominance. And so I'm going to give you some examples of what that looks like. And then if we have a few minutes, we'll talk about the other ones as well. But so over the week, we're going to look at all of these examples here. All right, so I'm going to start with incomplete dominance. So take a minute and read through that and maybe write some notes. So incomplete dominance is where you have a cross between organisms with two different phenotypes, but when they cross, it makes a third phenotype. So instead of taking a tall plant and a short plant and you cross them and they're all tall, in incomplete dominance, you take a tall plant, a short plant, you cross them, and now you've got medium-sized plants. And so what happens is you get a third genotype, or I'm sorry, a third phenotype, where that third phenotype is a blend of the first two. So if you look at the picture there, so we've got a red flower and a white flower, and we've crossed them. So if you've got a homozygous dominant with a homozygous recessive flower, red and white, you cross them. When you get the cross, you end up with a blend, a mix. And so it's pink. So in incomplete dominance, the heterozygous genotype always makes a blended phenotype. Does that make sense? And so then when you take that heterozygous big R, little r, pink flower, and it were to, maybe reproduce with another heterozygous big R, little r, pink flower, then you would get that 1 to 2 to 1 ratio. But now, again, every time where you have a big R, little r, a heterozygous flower, you're going to end up with a blend, which is pink. And so what happens is the dominant allele is not completely dominant. So that's why we call it incomplete dominance. It's not completely dominant over the red. So I'm going to take a minute and let me stop sharing of my PowerPoint and let me screen share my document camera. And so the thing with, can you guys see that now? Mm-hmm. Okay. So the thing with incomplete dominance is that we still use a big R or a big letter to represent the dominant allele. Let me turn that light off, see if it was better. So we've got the dominant allele, and we still use two little letters of the same type to represent the recessive allele. But that heterozygous, so the big R, little r, is always going to be a third phenotype. So we don't just have red and white, we don't have 1 and 2, now we have a third phenotype. So in incomplete dominance, the dominant allele is not completely dominant over the recessive allele. It tends to make this blend. So in incomplete dominance, you always get a blending. So if we were to do the same thing, and maybe we were talking about blue flowers. So we've got maybe the blue flower is dominant to a yellow flower. So if we take a blue flower and a yellow flower and we cross them, what do you think the phenotype might be here for this one?
It'd be green.
It would be green. It's just a blend. Okay. Now we work these the same way, like with Punnett squares, we work these the same way as we did the other thing. So we could take a green flower and cross it with another green flower, and we could fill in the Punnett squares. So we got big B, little b, big B, little b, little b, little b. And so the offspring here would be, what color would these big B, there's one of those, what color would it be?
Blue.
Yep. What would be the big B, little b? There's two of those.
Green.
They would be green. And then little b,
Yellow.
there's one of those and they would be yellow. So we see 3 phenotypes instead of just 2 phenotypes. And that's incomplete dominance. All right, does anybody have any questions on incomplete dominance? Okay, so I'm gonna go back to our PowerPoint and talk about the next, let me see if I remember how, there it is. Talk about the next type of pattern of inheritance, which is codominance. And just by the word codominance, what both, I mean, it kind of is what it sounds like, where both traits are gonna be dominant together. And so we're going to use our red and white flowers again. So in codominance, both traits are equally dominant, so both traits show through. So in codominance, if we use our red and white flower example, so if you take a red flower and cross it with a white flower, you don't get a pink flower now, you get red and white spotted flower. So both traits are going to show through. So basically 2 traits, both are dominant. They make a third phenotype, but that third phenotype is a hybrid because it's got both of those traits showing through. And if you look down at the bottom at the Punnett square, you see that we don't use lowercase letters now, and the white, instead of using a little r, because a little r would say it was recessive in some way, we're going to use all capital letters. So if both traits are dominant, both traits get a capital letter. So this is where we're going to use different letters instead of the same letters. So I'm going to jump back over to my document camera. Sorry, back and forth here. And we'll look at a few examples here of codominance. So with codominance, gosh, there's a weird glare always on this thing. I guess I need to move away from the window. Can you guys see that okay?
Mm-hmm.
Okay, so in codominance, I'm just going to use the same example. If we take the red flower and we cross it with the white flower. So in this instance, okay, the red is going to be dominant, big R, but the white is also dominant. So it's going to be big W. When we cross those two, all the offspring are going to be RW. So all of them will be spotted. So they're going to be red and white spotted. Okay. We see this in, we'll talk about blood types, but we see it in blood types. But we also see it like in fur color in cattle and horses. If you guys ever heard of roan. So roan fur color is basically a combination. So if you have a red cow and a white cow and you cross them, they make roan, which is red and white. And literally, like the hairs on them, you'll see a red hair and a white hair and a red hair and a white hair. And it's not spotted, but both of the hair types show through. So I'm going to go ahead and continue on here with this example here. So if we take 2 red and white spotted flowers, so if we take red and white spotted and we cross it with another red and white spotted, what would their offspring be? So again, we'll just do a Punnett square. And now we have different letters, but we set it up exactly the same. And so this is where we're going to start getting a little bit more complicated, where we'll have different letters and different superscripts on our letters here. But all of it is basically the same thing. So if each parent carries 2 alleles, but they happen to be different, we can still use different letters and it's all okay. And so here we've got RW, here we've got RW, and here we've got WW. So if we look at these phenotypes, we've got one that's RR, and that one would be red. And we've got 2 that are RW, and those would both be red and white spotted. And then we've got one that's WW, and that one would be white. So in codominance, we've got two alleles that are dominant. Both of them, if present, are going to show through and make that red and white spotted. It's going to show up in some way that they're both going to show up together. So just to quickly review, we've got incomplete dominance, where the dominant is not completely dominant over the recessive gene. And so you get a blend, okay? So with incomplete dominance, you get a blend. But with codominance, both of them are equally dominant, so you get spotted. Okay, so two difference there between codominance and incomplete dominance. All right, questions? So I'm going to go back to my PowerPoint slide. Let's see. All right. Is it going to let me move forward? There we go. Okay, so like I mentioned here, roan furred color in cattle. That's a really common example of codominance. So we can see a red cow and a white cow, and you get basically red and white spotted, and the hairs are literally, you have both colors of hair which show through. Okay, I don't want to get into polygenic traits today, but we'll come back to that on another day. I do want to talk about multiple alleles though. So, so at this point, we've only talked about where you have two alleles. You have dominant or recessive. You have incomplete dominant, where you can have, you know, this blending effect, or codominance, where both of them should show through. But oftentimes, well, actually more often than not, there are traits that are controlled by more than two alleles. So go ahead and write this down. When a trait is controlled by more than two alleles, that's considered a multiple allele trait. And so a good example of this is blood type. And actually, blood type is a little bit complicated because it's controlled by multiple alleles, and there's also codominance. But for simplicity's sake, just go ahead and write the definition. So multiple alleles is when there's three or more alleles. Okay, I'm going to flip to the next slide here. Don't want to talk about blood type yet. So let me stop my sharing on that. And I'm going to give you another example that's not blood typing, and we'll talk about blood typing later. And we've got a simulation lab to do later in the week on blood typing. But let's go ahead and look at an example of multiple alleles here. So 3 or more alleles for a trait. Okay, so in this case, we might have, let's talk about, there's this example of fur color with chinchillas. Okay, so we're going to use C, so they can be brown, or they can be tan, or they can be gray, or they can be white. Okay, so instead of just 2 versions, we have 4 different versions here. So 3 or more. So this is an example of multiple allele. So you notice here that I put, so we've got brown, and that's a capital letter. So that one's going to be the dominant, and that's the most dominant. And then tan, it's dominant, but then there's this recessive above it. And then here we've got CCH, which is gray. So it's more dominant than the white, but it's less dominant than the tan, and then the completely recessive white. So I kind of put these in order of what would be dominant to recessive. So what if I had a tan and I was going to cross it with a gray? Okay, so if I have a tan, it's going to have that C, big C, but it's going to have another allele. So if it was tan, it couldn't have the brown allele because that one's completely dominant. But it could have another one of these, or it could have one of these recessive alleles. I'm just going to say that it carries the recessive white allele. So if this is more dominant than this, which is more dominant than this, then this tan guy could have this more dominant tan allele and carry the recessive white allele. Does that make sense? Hopefully. All right. And then they're going to make a cross with this gray dude. And so he's CCH. We're going to say he's a purebred. He's completely gray. So he's got only that gene. So we've got a whole bunch of letters here. And so let's go ahead and make our Punnett square, make it a little bit bigger since I've got more letters to worry about there. So we're going to take the one parent and put it on the side. And we're going to take the second parent and put it across the top. Again, I set it up just like every other Punnett square. So each allele goes on a different box. We just have more letters to deal with at each time. Does that make sense of how I set that up? Okay, so let's go ahead and fill in our Punnett square. So we have big C, C, little c, CH. Big C, C, little c, CH. Little c, CH, and little c, CH, and little c. And so this one, the reason why I put the little c, CH first, you guys can zoom in that a little bit, is because you always want to put the more dominant trait first. And so this trait is more dominant than the recessive white, if that makes sense. So let's look at our offspring here. So our possible genotypes, we've got big C, C, little c, CH. And so there's two of those, right? So both of those are the same genotype. Did I do that? Yeah, I did that right. Okay, so what would this phenotype be? Would it be brown, tan, gray, or white?
Would it be tan?
It would be tan. So you're going to look at the most dominant allele. So what is that most dominant allele? And up here, so that says so two of these are going to be tan. Okay, and then these two are the same. So we have C, CH, C, and oops, no, I wrote that again. And so there's two of those that are exactly the same. Oh, I write that there. So we have two of those. So again, this trait right here is dominant to this trait. So here, CCH. Look at, so this is gray with the recessive white, so gray is more dominant. So these two are going to be gray. Okay. All right. So does that make sense on how to do a multiple allele Punnett square?
Mm-hmm.
Okay. So that's all the notes I want to go over today. But so 3 different variations. So let me go back to my, so we have incomplete dominance, where one trait is not completely dominant over the other, and you get a third phenotype that's a blend. So pink. So if you get 2 colors and they make a third color that's a blend. Let's see, I'm sorry, I heard somebody halfway talk. Does somebody have a question? I couldn't hear. I heard somebody say something, but I couldn't hear it clearly. Was there somebody who had a question?
I heard it too.
Okay.
I don't know what it was.
Okay. If your microphone's not working and you have a question, go ahead and type it and I'll answer it for you. Okay, so while you're trying to type, I'm going to go back over to this. All right, so if there's a blend, it's called incomplete dominance. So if you get 2 that's a tall and a short and you cross them, and then the third is medium, that would be incomplete dominance. Codominance is when 2 different colors or 2 different types show through. So maybe you have round hair and spiky hair, and you cross them and you get round or curls with spikes. So we'll say curly hair and spiky hair, and their offspring have curly and spiky hair. So that would be codominance, where both of them show through. And then in a multiple allele trait, there's just more variation. So instead of just having two, like brown and tan, now we have more variety, more variation, which just leads to more complicated Punnett squares. So instead of just two choices or three choices, we have more available possibilities of phenotypes. Multiple alleles is kind of how our eye color works. And so we'll take a little bit more look at that later. And if you want to scroll through the PowerPoint, you can. But our eye colors are multiple alleles. And that's one of the reasons why we have so many different types of eye color. Okay, any questions on anything? All right, so that's all the notes I'm going to go over today. But if you have that PowerPoint, if you want to pull that up and look at it later on, we're going to continue working through blood types, and we'll talk about polygenic traits and some other things along the way, but I think you've got enough for today. The only other thing I'm going to ask you to do today is there's a quiz on Canvas. So after you've taken your notes and kind of go back through and look at them, there's a quick little Canvas quiz. It's only 5 questions. And so you can do that quiz, and that's it for today. Tomorrow you'll have a more lengthy assignment where I'll ask you to do some reading and some practice with each of these types of traits. And then we'll continue on throughout the week. All right, does anybody have any questions? Okay, well, I'd love to see you all. If you have any questions, let me know. I'm gonna stop the recording.